Welcome, 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 everyone. We have a lot of new faces. Welcome to IDM Study Club. I love seeing everyone out there. Hey, welcome, everyone. Welcome all our good dear friends. Welcome our saliva hackers, our IDM clan, all the
Look
new
it.
faces.
There's Tribeca is here.
You see them, there they are. Hi, team. We got a lot of familiar faces. Some of our good friends are popping in. Very excited for tonight. I actually am going to, it's right at 8 o'clock, and I am beyond excited to have this webinar going on with our dear friend, Anne. But I am going to start off official because we're going to read her accolades. This is somebody, when I say you're about to meet the guru of brain health, I mean that from the bottom of my heart, of that we have to know all the amazing things that she has done and kind of hear from it. But I did tell everyone online, if you were following me on my Instagram or on Facebook, I told you to bring your cup of coffee, right? Because this woman is going to take such complicated information and be having a cup of coffee with you tonight as we go through study clubs. So grab your coffee, because you're going to want to be learning and taking some notes. But I want to introduce you to Anne Rice as I know her. If you don't know about her, I'm going to put my glasses on here, is that she has more than 30 years of clinical hygiene practice. She has witnessed the impact of periodontal inflammation on a wide range of diseases and conditions. She is a graduate of Wichita State University in dental hygiene. She earned a bachelor's degree in oral health and promotion, and she is a certified dementia practitioner and brain longevity specialist. I'm going to say that again. She is a certified dementia practitioner and brain longevity specialist, because I don't know anyone else who has that, what I'm going to say, accolades or criteria behind their name, especially as a dental hygienist. You're such a leader and an inspiration in our community. Additionally, she's worked as a consultant with the Weill Cornell Alzheimer's and Prevention Clinic. She's with the Florida Atlantic Center for Brain Health and the Atria Institute. She's enjoyed being included in international consortium that is diverse network of brain researchers, clinicians, and institutions who support Alzheimer's prevention. Anne Rice has developed in 2017 oral systemic seminars and help inform dental colleagues, yes, like you and me, and dental providers as well, and the public regarding all things oral health. She has dedicated her time and lots of education and has progressed, but her primary focus is in dementia prevention. She truly loves working with the mind, the body, and the soul. Now, I have to tell you, that is something I can't say enough about what you have accomplished, and I think a lot of us forget that, is that you being a friend. I have to say, the first day that I met Anne, I stepped out of an oral systemic seminar. We were in one. This is not her seminar. We were at a conference, so I want to set that aside. And you know how you're like in a meeting and you're like, I don't know if I agree with everything like that. And that's okay. We don't have to always agree. But there was some dated information. So I thought, you know what, I'm going to sneak out. And as I was sneaking out, somebody else was sneaking out. And I can't remember what exactly was said. But one of us said something, it's like, did she just say what I was thinking? It was like we were in each other's brains. We immediately kind of looked at each other, and she started speaking my language. When I say that, she was like, this research on amyloid plaques and on oral systemic health. And she was like spouting out research. I was like, girlfriend, you're my new friend. You are my new best friend. Like, you are speaking my language. And I have to tell you, and a few people are laughing who actually probably know Anne, know that that was spewing out of her mouth. But I instantly just was like, oh my God, I have to be friends with this woman. Didn't know anything about her. But I am going to say, I was just kind of blew my socks off when we met, of all of the knowledge you had. And when I say you are the leader in brain health, I mean that. In our dental community, you truly are. So I thank you for all that you've done and kind of paving the way. You are the dental hygienist paving the way for so many of us, and very inspiring. And I look up to you. So I know you always laugh when I say that, but I really do, because I may work on the other side and have worked on physician side and know a lot in what I say cardiology. Thank you to Dr. Pritchard and to Dr. Golder and all our physicians out there. But really what you're doing in brain health is so empowering. But I want you to share, Anne, you were just here in Florida, and these are the
I
things
was.
you do. So tell
Yeah.
us, what were you doing? You're trying to get warm, I think, trying to escape Texas, maybe during the cold. But I was trying
Yeah.
to get her to come see me, and she wouldn't because she was on the other
I
side.
was on the other side. Yes, that's true. I think it's funny when you tell that story because you've probably conveniently forgotten that I was probably lipping off as I walked out of that and probably saying something that under my breath to myself, and you probably caught wind of it. But when somebody does an introduction like that, it is really tough because I hope that the presentation can even live up to some of that hype that she gave me. But yeah, I was in Florida. It had snowed here in Houston. They had done a ground stop on Tuesday, and then thankfully I got out on Wednesday, and my meeting started on Thursday. But yeah, it was absolutely wild. And Florida was cold. Okay, it was
Yes.
50s. It might have gone down to like the 40s at night, and I'd left snow, so it was okay. But anyway, so on the East Coast side, who my mentor and my how I really got my start was Dr. Richard Isaacson. I'd written a paper in 2021 where I really kind of ponied up dentistry. And what I mean by that is I said for Frontiers for this journal is I said that we are an effective healthcare field in reducing the risk of cognitive decline, and that all the things that we do, from it's not just slaying the bacteria, it's diabetes management, all of the different parts to that. Anyway, Dr. Isaacson had started Weill Cornell Alzheimer's and Prevention Clinic years ago. He has now since left, and then I left with him. I did get to work there for a bit with him, but um, he read that paper and reached out to me, and I've been consulting with him now for, I guess my math skills will work out to about 4 years. Um, and he has really given me and a little bit of dentistry more of a place in the prevention of cognitive decline. And I will say we're not going to talk about pathogens tonight. We're going to talk about viruses, but no one should say, or we need to say at this point, is that periodontal disease causes Alzheimer's disease. So it's a complicated beast with inflammation and all different parts to that, which is, as I said, not today's discussion. But I did go over to Boca, which is where he is now. His brother Stuart is a neurologist as well, and they have a foundation there, IND. And if you go to the website, I do want to tell everybody this. They don't know that you're there. They give everything away for free. They don't charge for programs and any of that stuff. They do the research. So his brother is very into Parkinson's, and Dr. Isaacson is Alzheimer's prevention. But if you go to ind.org/learn, there is 10-minute modules by Dr. Isaacson, Dr. Kellyanne Niotis, who I worked with at Weill Cornell, and then also she went with Dr. Peter Attia, and then now she's back out again. And a Danny Angerbauer, Dr. Danny Angerbauer, who I'm with a little bit at Atria. But they have these little modules. So if you're a physician or you have somebody that wants to know preventive strategies, they're 10 minutes each, super easy, giving you all the information of all the different things that he has done at the clinic, at all of the clinics for all these years. These are the things that are getting results. We are getting less amyloid. We, proverbial, okay, they are getting less plaques, less tangles. They're getting improvement in cognitive deficits, and the research that comes out of there is just absolutely amazing. There were a couple patients that I saw when I was there. And then he actually spoke, this is kind of interesting, who he never speaks anywhere, but it was close to his house for an airway group that had a meeting kind of in the north side of Miami. And that was interesting, just about, sleep and airway and all of that and dementia prevention. So they're doing great things. They have equipment there that right now there's 7 in the world. You take very small amounts of blood, and within 30 minutes, you can process 270 data points. It is the most amazing high-tech machine. Lots of good things are coming out. I was not as optimistic. I don't think 4 years ago or 5 years ago, but I think that we're going to have great gains in this fight. Huge. I think that they will be coming out with an app. We're doing a lot with dried blood. We're going to be able to measure amyloid. We're going to be able to do a lot of markers at home. So um, enough about all of that. So yeah, it's great. I get to work with him, and I do the oral health part of the patient's prevention strategies, and it kind of weaves into everything else. Dr. Pritchard, of course, knows this as well, and Laura, is that we see when we treat the periodontal disease, all the markers get better, all the inflammatory markers, all the things that are causing heart risk and puts brain at risk. So when you're beating your head against the wall to treat that disease, just keep beating it and trying to get your patient to understand, because you're really helping. And I always like to say that with these patients, there's family members attached to them. So as much as that patient might not be your favorite patient, there's a family member like me or Laura or whoever that loves that patient. So anything that you guys can do is helpful. So today, though, I'm going to talk about viruses more.
Well, thank you, Anne. I think that's the thing. Not too many people. I say I have Peter Tia's book, and that's the closest that I can say that I've come to him. But to know somebody, you're my next closest thing. I get to say, "Oh, I'm best friends with Grace." Who
Yeah, no,
knows?
I get to. It's
All
crazy
of you
too.
are one person closer. That's what I want to say tonight. They're all
Yeah.
one person closer.
I get to see all the famous people charts and have meetings, and I can never tell anybody because it's all so private. People are people. People are not any different. They're all the same, but it's exciting, especially to be on the edge of what could be coming for all of us for help. So.
I love it. Absolutely love it. I know our focus tonight is on viruses. The last few people are coming in here. It is a hot topic, as you and I both know, as we're expanding in this oral systemic arena. Yes, a lot of is on pathogens, but it's really the interaction, right? They talk about this pathobiome, what's going on in the brain, what's going on in the vascular system, what's going on in the body. And it's just the more we learn that it's not just one thing, and we'll talk about that I always say it's not monotherapy, it's combination therapy. And that's really what viruses have opened my eyes up to. I think all of us really going through the pandemic, right? Realizing how much a virus can affect us. So I'm excited for you to kick it off. I know you have so much information to share with us. And so I'm going to turn it over to you to share your screen. And so we can dive deep into viruses. And then at the end, please, people, drop your questions in chat. We'll have a Q&A. Like to make sure that we're always leaving with really important things that we can take back to clinical practice. I also want to mention we did drop that link in the chat so that you can go to that site that Anne was talking about, and you can go learn there and see what's going on. So great to bookmark that and have that on your computer.
Okay, so can we see everything? Can you see my screen?
Looks great.
Okay, perfect. This is when how many viruses we have. We're going to learn that here in a minute. We're going to talk about HHV. We're going to talk about herpes viruses, and not all of these. We're only going to talk about more of a relationship, the ones with brain health, oral health, some of the things that we're starting to learn. And when I say starting to learn, when you look at the research, it didn't start. It's been around for a long time. Dentistry is kind of more catching wind to some of this, and not all of the data is crystal clear. And I have learned that just because you don't have the answers yet, if you do have some sort of data, it is going to lead us more in a better direction. So we don't have 100% of anything. We probably never will in science, but there's some good things coming out. Thank you, IDM, of course, for having me. And I talked about we're going to explore the viruses in the family of herpes. I'm going to traverse in between how it's relatable to the oral cavity. There's one little section about the gut. It's really short. I am going to focus a lot on HSV1, cold sores, which is so common because if we think the responsibility goes to a dermatologist to help our patients with their cold sores, that is not going to happen. In case if you've ever tried to make an appointment with a dermatologist, they're on their next cold sore before they can actually get in there and have any kind of help. So, and of course, I like to talk about the brain, so that's included in all this. As I put in all my presentations, is that this is not considered professional medical advice. So you don't get that from TikTok or Instagram or a Yahoo like myself that's with you on a Wednesday night. I don't know your genetics. I don't know medications that you're taking, but what I will tell you is that all of this is based in science. There's not really strategies that I'm going to be talking about tonight that is even affected by this. I will talk about antivirals. Many of you that are doctors, you know how to write a prescription and you know the right ones to write. I'm going to tell you what it looks like right now for some of the antivirals, but always consult with a pharmacist, or you doctors always look into your education too when you write these prescriptions for antivirals. This is traumatic. If you live in Florida, if you look at this, and Texas and Louisiana and anybody in the South, this is not what you'd like to look at. But I like to look at it now because I think this is very important for dentistry, and especially people that are involved in this group and some of the other groups, is that tiny events can cause major disturbances. And what I mean by that is 1/2 of a degree Fahrenheit on the surface of the ocean changes a tropical storm into a hurricane. And what I want us to examine and always consider is that single tiny changes that you do in your work can have significant and positive disturbances in your patients' lives. In that same vein, not doing a tiny disturbance could be very detrimental to your patients. Also, with these little incremental changes that you make, you are going to be more satisfied in your career. You are going to reap some amazing rewards of your mental health in keeping in this industry that we know is dentistry. If some of you all are in medicine, when you feel success, when we make these changes and have our patients more successful, everything is going to be better. There is something that I heard this years ago, and I don't know if it was on some stupid talk show or whatever. It was a husband and wife, and the wife was saying to the husband, if you aren't helping me, you're hurting me. And I'm sure it had something to do with the kids. Maybe he wasn't doing whatever he was supposed to do at home. But I think about that in dentistry. I don't think that we have this middle of the road where we're not doing anything. We always have to, whether it's putting out fires, but always doing prevention. If we're not really helping them in any way, educating them in any way, we could be significantly hurting them just by being uneducated or letting things kind of slide. The herpes virus, Wikipedia says we have 130 in this HHV family. I've read research that there's 200 relatives in this horrid family. 9 of them are with us human beings, right? We think about cold sores, right? When we think about herpes, we either think about cold sores or we think about genital herpes. And in dentistry, we always think about oral herpes. But there's lots in this family. 1 and 2 is oral and genital herpes. And then we have chickenpox, varicella zoster virus. That's going to be our 3. Epstein-Barr is our 4. Cytomegalovirus is 5. And then we have 6A, 6B, and 7. Those are going to be in our roseola virus family. They're really ubiquitous. 90% of adults have antibodies for both of those viruses. That is not the focus tonight, but I am going to throw this little bit out about the brain. 3 out of 4 cohorts are seeing that 6 and 7 are higher in those antibodies in Alzheimer's disease samples as compared to patients that didn't have Alzheimer's disease. But the argument here is that, is it just opportunistic? And that's what we always have to think about with viruses, because we really don't know everything about the brain. And I wanted to mention this earlier. I'll just put it in here. There is research that Dr. Isaacson is getting involved with, and this is all stemming from COVID. There was an actual patient that has long COVID and has some incredible repercussions of that. And thankfully, he's done a lot of funding in this. And there's going to be a huge research project that's done. It's longitudinal, that we're going to be looking at COVID, all of these herpes viruses, and brain health. My hope and prayer to this is that I'm at now about a 40% chance that we're going to put a little arm in this about oral health added to it. I've had a couple interviews. I just don't, we just got to get a little bit more money. And I'm hoping that then we will be able to maybe tease out some data about the oral health parts of this as well. So I'm kind of excited about that. But let's go back to, and Kaposi sarcoma, right? That's HHV8. That is rhabdomyovirus is part of HHV8. Now we've triggered different diseases, right? We have cold sores, genital herpes, and then we have chickenpox and shingles when it's reactivated, that virus when we're older. Mono is coming from Epstein-Barr, which, by the way, there's also various cancers that go into Epstein-Barr, Burkitt lymphoma, Hodgkin's lymphoma, non-Hodgkin's lymphoma, some stomach cancers, nasopharyngeal carcinoma. Kaposi is really kind of rare. Skin, lymph nodes, and other organs. It does have links to lymphoma in and of itself. We do have some ways to kind of treat some of the symptoms, but we can treat the cancers effectively. So if this virus is leading to these cancers, it's treated like a cancer. So we have success in that way. Vaccines, the only ones that are available, obviously, is for chickenpox and for the shingles virus. I think I'll talk a little bit more shingles when we get moved into Alzheimer's a little bit. It is intriguing that 8% of the human genome is derived from ancient viral sequences, and they are primarily remnants of retroviruses that have been integrated into our DNA millions of years ago. And the most notable example of that is HERV, which is human endogenous retrovirus family. Some of those sequences play a role in even regulating our gene expression, our immune response. They're found in every tissue of our body. It's in the blood, it's in the brain. It is absolutely, it's integrated in our genetic code inside of our cells. Viruses are really the most abundant organism on Earth. Often we associate them with disease, but they are also essential to our human health. Particularly the gut has about 10 trillion viruses that are mostly with bacteriophages. So the viruses have infected the bacteria rather than the human cells. They play a huge role in shaping our microbiome. They influence our bacterial populations, contributing to overall microbial balance in the body. How does it happen? How do we get the viruses and all the things that go with this? So typically it's going to enter, this is the herpes family, through mucosal surfaces. So your mouth, your nose, your eyes, your genitals, or through broken skin. Different viruses have preferences to the sites that they enter. The virus is going to replicate at the initial entry site, sometimes causing symptoms, sometimes the sores, but often the primary infection is asymptomatic. The virus infects sensory nerves, the endings at the entry site. The virus travels then retrogradely, so backwards along the axon of the sensory neuron to the nerve cell body. We call that the ganglion. That specific ganglion, depending upon where that entry site is. So trigeminal ganglion for facial herpes, sacral ganglion for genital herpes. So once it's in the ganglion, the virus will establish its latency. In this state, the dormancy state, where the virus's genetic material, the DNA, persists in the neuronal nucleus, but very few viral particles are produced. This makes the virus essentially invisible to our immune systems. No new virus particles are produced during latency. Then the virus does express some RNA. So those molecules called latency-associated transcripts, they call them LATs, are thought to play a role in maintaining that latency and preventing viral reactivation. So how does it get reactivated? Well, we get it reactivated. Is it stress? Is it illness, sunlight? When we talk about cold sores, hormonal changes, when we change in our immune system, normal fluctuations in our immune system. So when it gets reactivated, the virus then travels forward along the neuron's axon back to the skin or to the mucosa. So the virus replicates at the original site of that infection. This leads to the production of the new virus particles. Now we get the symptoms. Now we get the sores, the blisters, so on and so forth. Viral shedding can occur even in the absence of noticeable symptoms. The virus then can cause recurrent outbreaks near or at the same location. Hello, cold sores. The frequency and severity of the reoccurrences is very varied, depending upon the individual. So these have co-evolved with humans for forever, for a long time. Establishing the latency, reactivating after, and that is kind of the key to how they've had this long-term survival and success. So they don't mutate, but they can change a little bit. They also don't do it like rapidly, like say an influenza virus is going to. There are some genetic changes that are happening over time, but they're pretty stable in the genome, and that maintains their strategy. I will tell you that talk about invasion, that is my dad. That was World War II on Ewo. There he is. So, okay. Moving on. I'm not going to focus on chickenpox, varicella zoster very much. I'm just going to throw a few things in here. That chickenpox, that's the only thing that we have a vaccine for, and then we do have shingles. Chickenpox can actually lead to encephalitis, shingles, that reactivation at later in life, the chronic nerve pain. I don't know if anybody's had it. It can actually lead to stroke and meningitis as well. There is great research. Chen and Bay both did, and it's very small on your screen, but shingles is associated. It's a clear, but it's a small risk for increasing the risk of Alzheimer's disease and dementia. Why is that? Well, it's in regards to neuroinflammation or damage to the blood vessels. I will say that at the clinic, I'm not your doctor, but the recommendation is that everyone gets a shingles vaccine. In the elderly population, we don't need a reoccurrence of any herpes viruses. That includes our cold sores and everything else. So shingles vaccine is probably a good option for many. Oral tissues, you could have the ulcers there, right? There could be a dry mouth because we're going to have a dysfunction in our salivary glands. That nerve pain, we might see this in dentistry. That could be mistaken for a toothache for people that have chickenpox and shingles. And I had chickenpox as an adult. I didn't get it until I was almost 50 years old. It was a lower version of it, but it was kind of overwhelming there. Okay, HSV1, cold sores. This is where we're going to spend a little bit of our time. A person with HSV1 can transmit the virus even though you've never had a visible cold sore. So that is why most people have this, the virus, before the age of 5. This occurs asymptomatic viral shedding. It's present on the skin or in the mucosal tissues without those noticeable symptoms. Obviously, you could get it through kissing and sharing utensils. And whenever I see somebody drop the binky on the ground and they put it in their mouth, hello, HSV. So there we go. Because of that asymptomatic shedding, most individuals that have it are completely unaware. I get into this in a couple of different slides, but I'll mention it now. Dr. Ruth Tataki has studied cold sores and brain health for, I think, 3 decades now at this point. And there's some fascinating things. She was considered an outlier, but now, of course, she comes into the fold. And any of you in dentistry know that some of your outlying ideas tend to come into the fold at a certain point. But she talks about that 80 or 90%, whatever the percentage is of us that have the cold herpes virus cold sore, isn't it interesting that only a fifth of us actually get the lesion? And that's going to lead us into some gene play when we talk about reactivation, asymptomatic, and laying dormant. So the ability, it's rather remarkable that you can have lifelong latency and then you have not had an occurrence of this. So it's in that trigeminal ganglion, and it's located near the ear. It's kind of a sanctuary for the virus. It shields it a little bit from the constant surveillance of the host immune system. Now that trigeminal ganglion, you've been in dentistry, took some classes, innervates the face, lips, oral cavity. So that's that direct pathway to the replication and to the shedding. When we talk about the gene interplay in this, bear with me just a little. I don't know if this is kind of boring to some of you, but the way the host genetics works. So we have variations in all of our genes. What you have, what I have, your sister, mother, there are variations. And how your immune system responds to that is very different, including with HSV1, and how effectively it can keep that virus in that latent stage. So some people may have genes that make them more susceptible to reactivation, or they're less efficient at suppressing the virus. There are some studies that suggest that people who carry, there is a gene and a variant, APOE, is the gene. You get one from mom, one from dad, and then there's different alleles or variants. You get a 2, 3, or a 4. 4 is your susceptibility gene to Alzheimer's. Make no mistake, plenty of people get Alzheimer's without this gene variant. This just increases susceptibility. 3 is considered neutral. Plenty of people have Alzheimer's that are a 3. And 2 is considered protective, but we are seeing some results that 2 and Parkinson might have some relationship there a little bit. But so this APOE4 gene variant, you may be more susceptible to developing cold sores. There was a study back in 2010 in Infectious Disease Journal. People with this variant are more likely to experience recurrent oral herpes outbreaks. Not everyone with this gene, right, is going to develop cold sores. So there's viral genetic. We have different strains of HSV1. Some variations in the genes can affect the ability to establish latency or to reactivate. Some strains might be more prone to reactivation than others. There's genetic studies that suggest that HSV has multiple genetically diverse lineages that has evolved over time. We look at in whole genome sequencing at least 6 major phylogenetic types. So when we look at places like Europe, Asia, Africa, there might be different types in there. Gene expression. So the expression of certain genes, both in the virus and in the host, can influence the switch between latency and reactivation. The expression of, it's called latency-associated transcripts, or LATs, in the virus help maintain that latency, and that can be different depending upon the person. When this is providing this safe haven for the virus, it does necessitate a periodic reactivation to ensure its propagation. So the triggers, the hormones, the trauma, the UV might work for you or might trigger for you and might not trigger for somebody else. So the virus is traveling down that nerve axon from the trigeminal ganglion to the skin, mucosa membrane, the mouth. That journey along that sensory nerve pathway is the characteristics of the prodromal symptoms. So if you've ever had cold sores, you know it before it's there, right? That tingling, itching, burning, there's lots of different ways that people describe that. So that is it going these sensory nerve endings before it's going to pop up. It replicates in our epithelial cells, causing the characteristic vessels, those lesions that we see on our faces or wherever it's going to land. The interplay between those, between HSV and the sensory neurons, is not really passive. It actively manipulates the neuronal environment to ensure the survival. So it can evade apoptosis, allowing the infected neuron to persist despite the presence of the viral DNA. So the virus can modulate the host immune response, preventing complete viral clearance. When I talk about it being contagious, I have not met a hygienist that has gotten herpetic whitlow. We know that in healthcare and we know that in dentistry this is possible. This is a beast of contagion, especially when the vesicle is there, right? So we know the viral shedding. We know that we can get it that way. But I want to talk about the cornea here a little bit because this is a family dynamic. So we can get that herpes inside of our eye and we can have cornea scarring. That chronic inflammation can cause something called stromal keratinitis, where the deeper layers of the cornea are damaged. This is my husband, who doesn't have vision in one of his eyes. He was a kid, he got it in his eye. And the only solution to this is a cornea transplant. Also, you can get this virus up your nose. So in all of these cases, this has relevance to dentistry. I wouldn't think that you want it on yourself, and I don't think you want to give it to your patient in their eye or up their nose. We need to be very protective. I will talk about aerosols and all of that. I'll talk about lasers and get into that, but we need to be really cautious with this. You know, we use eye protection, and I've been in the offices and I've given out that eye protection, and the women that wear the eyelashes complain because it falls and hits their eyelashes. So then when you pull it out, there is this opening, right here. So there's a lot of aerosols that can come up that way. Always have them close their eyes, or get a little bit more protection on people's eyes. And then up the nose, I'll get into some nose filters here in just a little bit. So one of the major reasons it's so challenging to demonstrate that a virus can directly cause neurodegenerative diseases is the timing. Some viruses, herpes simplex 1 or cytomegalovirus, can lay dormant in the body for years and years and years before reactivating. So it would be difficult to establish a clear timeline between the initial infection, the onset of symptoms. On top of that, our immune systems are highly individualized. So not everybody exposed to the virus, right, is going to experience the same symptoms or the long-term effects. We do know this about cold sores: oxidative stress. HSV1 elevates oxidative stress, and that is strongly implicated in Alzheimer's disease risk. We do think that it's the reactivation that is the critical component in the brain. The likelihood of developing Alzheimer's disease in some of the research is 12 times greater for an APOE4 carrier who does have HSV as compared to having neither factor. So it can invade nervous system cells, the central nervous system, your brain and your spinal cord, right? Disrupt brain function. This is based on research about the host gene response, and then the neuroinflammation. So we know that the chickenpox shingles virus can trigger that brain inflammation, and it does damage neurons over time. There's over 150 publications talking about this relationship between Alzheimer's disease and HSV1. It's neuroinvasive. We know that it can culminate into the development of Alzheimer's disease. And I talked about Dr. Ruth Itzhaki, who her hypothesis is she feels that it's responsible for 60% of Alzheimer's cases. As I said, she's an outlier. I don't know that we're totally quite there yet. That's really difficult to do that cause relationship, but it's about the repeated reactivation. So this is where we really have to understand a few things. What we know is when we put people on antivirals, not only do you reduce the risk, you actually, not only do you not have the elevated risk, you can actually reduce the risk. I'm going to tell you a very generic story about someone that is in our world that has neurocognitive issues. And in this person that already had neurocognitive issues, they are giving this person, and they have for many years, antivirals every day to help them with inflammation. Is that what the recommendation is now? Do we put the world on antivirals to prevent this? No, that is not the recommendation. When you have the tingle, to get on that antiviral as fast as you can. If you have cognitive deficits, that is probably going to be the recommendation from the neurologist. But to have it as a precautionary antiviral, that is really not where they're going at this point. Let's talk a little bit more about Alzheimer's disease, and I'm going to talk about neurofibrillary tangles and amyloid beta plaques. So this is the hallmarks for Alzheimer's disease. So amyloid protein and tau pathology. It's hyperphosphorylated tau. We're going to do tau first here just a little bit. Tau is a protein that plays a role in stabilizing microtubules, which are part of the cell's structural framework, so the cytoskeleton. These microtubules act like highways inside nerve cells, inside neurons in your brain. It's going to get our nutrients, it's going to get our signals, it's going to get all the materials that are needed to make that cell function properly. Tau binds to those microtubules, keeps them strong and all organized. In Alzheimer's disease and other neurodegenerative disorders, tau becomes abnormally modified, phosphorylated, causing them to detach from the microtubules, destabilizing that cell structure, and it leads to clumps, neurofibrillary tangles. That's what we call that. It interferes with neuronal function and eventually can contribute to cell death. I will say tauopathy is involved in all neurodegenerative diseases. Parkinson's disease, there is tauopathy there. That is a different protein. It's alpha-synuclein. But this dysfunction in tau is most neurodegenerative diseases. So the physical or molecular triggers that these hallmark pathologies are associated with viruses, bacteria, fungi, parasites has been postulated for a very long time. One or more of the pathogens can trigger these neuropathological events in Alzheimer's disease. The aggregation of amyloid plaques. Amyloid plaques are there to try to sequester inflammation out of our brain, but leading to this tau phosphorylation, neuroinflammation, and then that neuronal injury. So in vitro and in vivo studies, it's supported that the brain's immune response as a mediator for the development of Alzheimer's disease pathologies. There's a reason why I had to give you a whole entire history of tau when we come This was research that just came out in January of this year. So tau may be protective in viral infections. So what they're finding is that they used little miniature organoid brain models. So they grow little baby brains in labs. And they found that the HSV infection increased tau levels. What was interesting about that those modified tau appears to actually lower herpes protein levels and reduced neuronal death. But then there's the caveat, right? Then we have tau that's becoming phosphorylated. So it led researchers to investigate the immune response pathway. How is this working in the brain? This isn't interesting to many, but we need to understand some of this. It's called a C-gasting pathway. What this is it detects misplaced DNA inside that cell, and it plays a real big role when you have cancer and fighting infections. So they found that this immune pathway is active where this HSV-1 virus is, and modified tau are present. So what we're seeing, what they're trying to get this relationship, the tau is trying to reduce this viral HSV-1, and then the tau then becomes corrupted. It's our brain's immune defense system. And we really haven't, up until this, seen exactly with HSV-1 how that tau actually works. This is going to be great things. Do we use perhaps antivirals, maybe a little more aggressively? This is Kellyanne Niotis, who was with Dr. Kellyanne Niotis, who was with us at Weill Cornell, and Peter Attia. She is down at IND. She's still, you will find her on the website and such, but unlikely that a single virus is solely responsible for diseases like Alzheimer's or Parkinson's. They act as contributors, exacerbating pre-existing vulnerabilities. It's all about risk modification. It's all about reducing risk. So there's lots of challenges in the pursuit to a cure for getting rid of HSV-1. Because it enters that dormant state, right? It's evading the immune system. It can evade actually antiviral drugs as well. So it periodically reactivates, leading to these new infections. The eradication is virtually difficult because it stays in our nervous system. Another big obstacle is our blood-brain barrier, semi-permeable membrane that keeps errant pathogens and things like that. It restricts the ability of a lot of drugs to reach the virus that's inside the brain, inside those neurons. So it requires treatment somehow that we have to figure out that can cross the blood-brain barrier without causing toxicity. That's one of the biggest problems with the brain. So even existing antiviral drugs like valcyclovir, it's effective at controlling outbreaks, don't eliminate that virus, right? So we have evolved mechanisms to evade the immune system, the interference with the pathway that I mentioned before, the gas sting, preventing the body from fully clearing that. So we do advanced technologies, gene editing, CRISPR, even though in this case, CRISPR, they're doing something called meganucleases. Those are two different things. So to eliminate the latent virus, but they have to figure out how to deliver this safely to the neurons because you don't want to have genetic damage. That remains a big hurdle. There is hope for this. And this has been going on for over, I think it's in year 12 at this Fred Hutch Cancer Center. So the herpes that is so sneaky, hiding, reawakens for those blisters. So there's experimental gene therapy. They're injecting inside the blood a mixture of gene editing molecules that seek out where is the virus that resides in the body. It's that mixture is included in that is lab modified viruses called vector, commonly used in gene therapies. There's also enzymes that work as molecular scissors. So once that vector reaches the clusters in the nerves, has the herpes virus hanging out inside there, the molecular scissors snip away at that virus, damaging them or removes that virus entirely. They've been able to do this. They cut it enough that it can't repair itself. And then the body's own repair systems recognize the damaged DNA as foreign, and then it dumps it. So they've been working on this. They've been able to eliminate it 90% of HSV-1 after a facial infection, and 97%, this is what I wanted to remind you, is that 90% of HSV-1 in a genital infection. So you can get HSV-1 also in the genital area. It took about a month. They're also working on adapting the same technology to target HSV-2 infections as well. So what are we going to do until the molecular scissors decide to cut all this apart? we're going to do antivirals. So acyclovir has been the first line of treatment. It's disadvantageous in some ways because it has low bioavailability. What does that mean? It's less than 30%. It means that you got to take it more. You got to take it more dosage. You got to take it for a longer amount of time. That is why valcyclovir was invented, to overcome that limitation. So the bioavailability is about 54%. So if you know about valcyclovir, we're not taking a ton of it, right? Now we have pencyclovir or famciclovir. This is what my husband takes. Bioavailability is at 77%. He takes a huge Mac Daddy dose, not in one pill. It's a single dose when he gets that tingle. So what we know is that we know that herpes infections can increase your risk of dementia, but the use of antiherpetic medications is associated with a decrease in dementia risk. You've got to write prescriptions for every one of your patients. If that's not on your health history that they get cold sores, and this doesn't matter, and I say this, and this is crass, but it doesn't matter that it's a hot 21-year-old. It matters in a 60 and 70-year-old and an 80-year-old. That is critically important that we are writing these antivirals for an older population. It has nothing to do with what it looks like. It has to do with reducing the replication and healing faster. This is an incredible drug. It's called Sitavig. I don't know if anybody's heard of it. You can tell me when I log off of this. But so Sitavig, it is a mucoadhesive buccal tablet. You're going to put it in the fossa of the canine. So if I have a cold sore on this side, obviously it's going to go up here on the other side, right? This has great results. It slowly dissolves 12 to 24 hours. There has been issues that it stays stuck where it needs to be, but I think that if you really wanted it to work, you could. And when I tell you the cost, you're going to figure out how that's going to work. They've had great results, complete healing. The patients that had gone on like 150 days extra without having a reoccurrence. It's not established in pediatric patients. And of course, there's no data on pregnant patients. But this is for a different group of folks. That's the cost for 2 tablets. You only need one. You get to keep that other one. But that is not possible for the public at large. It's a great drug. I am not quite sure why it's so expensive. There's good research. There's a valid trial. It was a first randomized double-blind placebo-controlled study. Does valcyclovir actually slow cognitive decline? That's the answers we want to know. This ended just in December. And what we're hoping is that we will be able to move this to a larger phase 3 trial. And these are patients, when you hear somebody that has mild cognitive impairment, that's kind of a garbage pill term, as my boss likes to say. That means we don't know why they're forgetting things. That's like this general term. We need to get a diagnosis out of that. So MCI could be from a lot of different reasons. These people had mild Alzheimer's. It was diagnosed. So this will be, I hope the results are great. So they were taken, and I thought that part of this had an arm of postmenopausal women, but I don't know that that's the case as well. But anyway, this will be great if it can get to phase 3 trials. So could you use a laser in your dental practice? They don't eliminate the virus, but it is going to slow the replication, reduce the pain, the duration, and the interval reoccurrence. Now, some of you all can tell me not every state a hygienist can use lasers, but every dentist can. That's for sure. We have all different kinds of lasers. I'm not a laser guru. I'm just going to kind of go through all the different laser, just a random, what you see on your screen. Every company is going to tell you that their laser is the only one that will work for cold sores. That is not true. So you don't have to buy a $40,000, $50,000, $100,000 laser. You can go basic, a diode laser, tabletop, $2,000. You can be able to do great things with cold sores. It has portability. The price tag is attractive. There's pulse dye lasers too. It's really not widely accepted. It's minimally invasive. They say that it's less scarring. They use them for aphthous ulcers and lichen planus as well. It is not overall across the board. I think most dental hygienists have diode lasers. I'm going to go over a little bit about how it would work. And if you do lasers, you already know how this works. But this is also considered what we call cold laser. And because it's going to deliver light energy, stimulating tissue healing without generating significant heat, making it minimally invasive. So How would you do this? You're going to start with a power setting of a 0.3 watt. You're going to hover over about 2 millimeters. Are you touching the lesion? No, you are not. You're going to move it cross-hatching motion over the lesion without touching it. You're going to move the laser energy horizontally, then vertically, cover the entire area. Then you're going to go to a power setting of a 0.5 watt. Then you're going to do it again, 0.7, do it again. You're going to finish up with a 0.9 and a 1.0, that cross-hatching constantly. Small lesion may take you 20 seconds per setting. Larger could take you 60 seconds. This is a 5-7 minute, maybe 10-minute procedure in your office. Many of you have an overflow room. If you can create an environment with your patients where you tell them, listen, if you get that cold sore, I am more than happy to help you with the healing process. We can do this in dentistry. No, I don't know that dermatologists do this. This is a great thing. I know that hygienists are rolling their eyes going, I don't need somebody on my schedule. How are we going to do this? You're always going to do this in an overflow room, right? It only takes a few minutes. You can send somebody else in there to clean up all of the things, and you can go back to your operatories. This is a good friend of mine. I don't get anything from this, but she is the guru for lasers. Her name is Angie Wallace. I worked with her 20 years ago. She happens to be from where I'm from. Only about 25 hygienists in the world have advanced proficiency in lasers. She's a master with the Academy of Laser Dentistry. She does in office. She can answer all your questions, and she will answer every single email that you send to her. Now, there's lots of precautions should you decide to use these lasers in your practice. The Occupational Safety and Health Administration, there's nothing to tell us about what we're going to do about the plume. But what we do know is that this plume, the smoke or the aerosol generated with that laser energy, that when we vaporize the tissue, it can contain cellular debris, bacteria, chemicals, particulates. I know technically you aren't touching the tissue, but that energy that it emits can interact with the air itself, causing some degree of ionization and small particulates. The energy could be reflected or scattered, even if the laser does not touch the targeted tissue, could be close enough to maybe ablate or vaporize small amounts of saliva or blood. That high vacuum, I mean, that it needs to be so strong. You're going to do that while the laser's firing at all points in this. You're going to keep the site cool to remove that plume. Nose filters can be added to this for the patient and for you. Remember what I talked about, the nasal herpes, right? That protective equipment. Nose filters are lightweight, they're discreet, they're latex free. They have multiple sizes. In 2 slides, I'm going to show you a little bit more about that. Your mask, are you going to wear a level 1 mask? Well, no, you're not. You're going to either do an N95 or you're going to do something that's going to filter out most viruses. And then you're going to control that plume as well. Obviously, no reflective surfaces in the operatory. You know all the rules when it comes to lasers, the metal, the mirrored surfaces. Have we thought about earrings? Earrings on your patient or earrings on your head? That's another area. And then you have to be properly marked, the safety zone, the 8 feet. Let's talk about those particulates a little bit. So the herpes virus is about, HSV1 is about 120 to 200 nanometers in diameter. That's in its virion form. So when it's aerosolized during procedures, when you decide to clean somebody's teeth or your doctors are going to do fillings or some sort of treatment, ultrasonics, air polishing, the high-speed hand pieces, it contains, the virus gets into the saliva, right? The droplets, and then we get bioaerosols. How big are they then? So those large droplets are greater than 5 micrometers. Typically, those will settle quickly, but then we get the fine aerosols, right? They remain suspended in the air for extended period of time. HSV is super small, but it can transmit through the aerosols because it's attaching into the saliva or to the blood or to whatever you're doing. Some people decide to treat, we just don't recommend it. When I have a husband that can't see out of one of his eyes, I just don't want the virus going around the room. Plumes are very controlled. You're doing the laser, you're stopping replication, you have lots of a controlled setting. You have the HVAC, all of those things. Remember about viruses entering the respiratory system from those aerosolized droplets. They are breathing through their mouth, they're breathing through their nose. This is a little bit about nose filters. I got turned on to these at about the COVID time, probably because I was still having to travel. I think this company is actually O2 nose filters. If you were to Google them, I don't get anything from this, but it filters about 90% of airborne particles. PM means particulate matter with a diameter of 10 micrometers smaller. They can go to particulates of about a 2.5 at about 65%. It's a little screen, blocks the path of the particles. You can still breathe through it. It acts like little magnets. It retains those particles while allowing the air to pass through that so that they're not getting it up into their system. We could use these. We could give these to our patients to protect them a little bit as well. Let's get into the viral influence on periodontal health. I'm already at 8 o'clock. Okay. The association between herpes viruses and periodontal disease. So viral DNA has been detected in our gingival tissues, gingival curvicular fluid, subgingival plaque. Cytomegalovirus has been found in gingival curvicular fluid from periodontal tissues, suggesting a bit in this research that the replication in these sites may trigger disease activity. So the oral cavity, we have a diverse viral community in there. We have bacteriophages that infect the bacteria, and in periodontal disease, that viral balance is disrupted. So it could influence bacterial composition, the behavior, contributing to perhaps disease initiation and progression. So certain viruses, especially Epstein-Barr virus and cytomegalovirus, are commonly found in patients with severe periodontal disease. It weakens local immune response. It also allows those pathogenic bacteria, PG, TF, to proliferate, exacerbating that tissue destruction. There's an immunosuppressant with this, right? We're not going to have a great as immune response, which is not going to help in our periodontal disease issues. Increased inflammation. It triggers and amplifies inflammatory responses. And then that is going to be triggering inflammatory responses in periodontal health. That persistent inflammation, tissue breakdown, bone loss, all of those things with periodontal disease, and directly impacts perhaps host cells. We do know cytomegalovirus and Epstein-Barr virus can infect periodontal cells, including fibroblasts and epithelial cells, leading to an altered cell function, increasing the susceptibility to a periodontal breakdown. So the oral gut-brain axis, so we swallow bacteria all the time. They're live, they're dead, they're all the things. We know that some of the bacteria can survive stomach acid. We do know that there's a concept of this oral gut-brain axis, this communication pathways that link the mouth, the GI, and the central nervous system. Viruses can disrupt, first of all, the gut microbiome, leading to a dysbiotic environment. It can enter through swallowing the virus-laden secretions, right? It can enter the enteric nervous system as well, reaching the vagus nerve ganglion. The vagus nerve here up to here. Some studies suggest that reactivation of latent herpes virus is correlated to pro-inflammatory gut bacteria, worsening IBS, IBD, and leaky gut. There was a small study, it's not very big, but patients with IBD had a positive relationship to not only cold sores, HSV1 and 2, cytomegalovirus, and varicella zoster virus. So this is something also to consider with IBD patients. What do they take for IBD? They take an immunosuppressant. That is going to increase your risk of developing opportunistic infections. There is a thing called HSV colitis. Corticosteroids increases the risk of reactivating dormant herpes infections. So poor oral health, untreated oral diseases with the mouth can affect the gut and leading to neurodegenerative relationships with this. We do see some chronic fatigue syndrome patients have reactivated herpes infections, and they have gut dysbiotic environments and gut permeability as well. Cytomegalovirus, double-strand DNA virus, integrates into host cells, can lay dormant, not cause a lick of problems, but in an immunocompromised individual or can cause some severe diseases and disruption. There is a relationship between cytomegalovirus and glioblastomas. It has not yet, as Epstein-Barr has, been classified as a definitive carcinogen. It has lifelong latency, right, in those host cells. It is neuroinflammatory. It does disrupt the blood-brain barrier and can alter the immune response in the brain. New research out of, this was not, I think it was last fall maybe, Arizona State and Banner Alzheimer's Institute suggests that this cytomegalovirus contributes to Alzheimer's disease. Even though typically dormant, it can remain active in the gut, traveling the brain. Remember what I talked about on the other side through the vagus nerve. 90% of people develop antibodies for cytomegalovirus, and this is funny in this research, by the age of 80. Okay, by the age of 80, we're all going to have antibodies for it. Usually, as I said before, it is no harm, but we do see persistent cytomegalovirus activity triggering an immune response that promotes those amyloid plaques and tau tangle formation in the brain. You might hear when people talk about Alzheimer's disease, they talk about CD83. That's what I'm going to get into a little bit. Cytomegalovirus in the brain leads to an activation of microglia. These are your immune assassins. They're lovely little folks until they get overstimulated. Then these microglial cells cause neuroinflammation. If they get overactive, this chronic inflammation causes neuronal damage. This virus triggered microglia to express a gene, CD83, and that is linked to Alzheimer's disease. There is this discovery, which is great. This goes back into antiviral drugs, perhaps, maybe slowing the progression if we find this in the patients, if it's activated, so on and so forth. The little brain organoids again, same research. So they made these little brains, and cytomegalovirus increased the production. They were able to grow it, amyloid plaques and phosphorylated tau proteins. It's great support that they're going to do lots of investigation, which is a little bit of why they're doing this intensive study. Dr. Isaacson is down at the clinic. So suppressing it with antiviral medication. Cytomegalovirus is not the same as acyclovir and valcyclovir. You can read on your screen all the different ones, why they use it. It is not, as I said, it's not the same. Are you going to put a patient right now on an antiviral? No, you're going to leave that to their physicians to decide. Oftentimes they use these in really immunocompromised transplant patients, or people that are, excuse me, some resistance people that have kidney issues. There's issues with that as well. So it is different. Acyclovir doesn't cover all the bases. Clinical manifestations that you might see: oral ulcers. You might see it on the tongue, the palate. They could be necrotic lesions, irregular borders. They could be shallow. You might see this more with our HIV patients, transplant recipients, or somebody that's on chemo. The periodontal involvement, we talked about that. Xerostomia, there could be some salivary gland issues that are happening from this as well. Candida, which is going to be the secondary fungal infection from this. And then when I talk about the gland infections, you might be able to see this with your patients, that if they have swelling, if you have a patient that comes in, their salivary glands are swelling, they're uncomfortable, they have a reduced salivary flow, maybe it is an exacerbation or a cytomegalovirus rearing its ugly head. In periodontal health, I kind of went through this. We do know that these infections are stimulating certain inflammatory molecules, tumor necrosis factor alpha, your interleukins. We know that AA, that is associated with aggressive periodontitis, even though we all know that there's AA and not aggressive periodontitis. Cytomegalovirus has had an association with AA a bit. So there is that relationship. And when we talk a little bit more about Epstein-Barr, we're going to see AA and PG rearing its head. It infects epithelial cells, immune cells, monocytes, and T lymphocytes also. So Epstein-Barr double-strand DNA virus, it integrates into the host. We know this as mono, right? It's a group 1 carcinogen. I talked about some of those in the beginning with Burkitt's, the lymphomas, nasopharyngeal carcinoma. There are lots of implications in not only Alzheimer's disease and Parkinson's disease, but also in multiple sclerosis. Studies have proved evidence that it is a cell cycle dysregulation that plays the progressive role in the neurodegenerative diseases that are altering the way that the entire cells are working. So we do see that also with Epstein-Barr virus, it's going to move into sort of Guillain-Barre syndrome. I have a brother-in-law who Epstein-Barr, and then now he has Guillain-Barre syndrome, and it's not great. Research is suggesting that the Epstein-Barr infection increased risk of developing, and the nature of the relationship is kind of being investigated. So the way that it might work is EBNA is a protein. It is very important for Epstein-Barr survival and persistent in that host. It's consistently present in latent infected cells. So it could be a target for the immune system. So the presence of antibodies that target this protein, EBNA1, indicates the immune system has encountered and responded to the Epstein-Barr virus. So these vesicles, the tiny particles released by cells that carry various molecules, including proteins and antibodies. Researchers are now investigating these vesicles in blood samples carrying antibodies against the Epstein-Barr. I'm sorry if you can hear my dog. The Epstein-Barr antibodies, they could potentially serve as biomarkers for diagnosing an Epstein-Barr infection, assessing the activity, and even understanding perhaps the role it's playing in the Epstein-Barr virus. So they're looking at the antibody relationship with the Epstein-Barr and then that for Alzheimer's disease. There could be a direct infection of Epstein-Barr into the neurons, the immune system modulation, the multiple sclerosis, as I talked about before. Epstein-Barr virus infection precedes both the first neurological symptoms and the early signs of MS. So with all of the neurological diseases, Alzheimer's, Parkinson's, Lewy body, there is this relationship between Epstein-Barr, and that is probably more of a topic of conversation, more so than cytomegalovirus, I would say. Epstein-Barr, there's really no antivirals approved to totally eliminate it, as we all know. In severe or immunocompromised cases, sometimes they do antivirals a little bit off-label. So we're going to do our acyclovir, valcyclovir, but you're going to see a cytomegalovirus antiviral, that ganciclovir is in there. And then if there's drug resistant, they have to go to another antiviral, but it's not used all the time. This is my kid who got it at the end of last summer. And you can, I don't know how well you can see that rash that he got, it was unbelievable. There was really nothing that we could really do about it. When are they used? We're going to severe infections that come from mono in life-threatening conditions. There wasn't a whole total lot that we're going to do with mono. You have to kind of wait it out. And then now I know all these things about mono. So now I'm paranoid for my son's rest of his life. Other oral manifestations of Epstein-Barr is oral hairy leukoplakia. Remember that this you can't wipe off. It's not like thrush, very definitive differences. Epstein-Barr being the primary cause of mono, 95% of Americans have this infection, but we didn't get mono. We get it by about the age of 35. Sore throat, tonsils. I hope you never have a patient that comes in with it and just said, oh, I have a little tickle in my throat. We do know that it's in periodontal pockets. We've been able to find it there. Causing those oral ulcers, salivary gland issues, swelling. Very similar. Epstein-Barr is similar to the cytomegalovirus. Epithelial cells and B lymphocytes within these gingival tissues as well. A meta-analysis really confirms this association between Epstein-Barr and the risk of periodontal diseases. This is probably going hand in hand, and this is where we get into AA. AA is associated with Epstein-Barr. And then on my next slide, I'll talk about Prophmonas gingivalis. AA can actively reactivate Epstein-Barr, which is fascinating. And we'll talk a little bit about it when we talk about how it works with PG. And then I'm at my very end of my slide. So, okay, we know PG, major player. Everybody likes to study it, right? It's been shown to interact with Epstein-Barr, contributes to the reactivation of it through different cellular mechanisms. And why is this? It has to do a lot with butyric acid. There's other ways in the cellular environment and different molecules, but I'm going to kind of explain. We think about PG in such a simple way, but it does so many other things to the body. So I'm going to try to explain this. I worked on this to be able to explain this easily. So PG produces butyric acid. So that can mess with butyric acid, can mess how your cells work. Specifically, something called histone, which is really not that important to this, deacetylases, which we're going to call it HDACs. So They're the gene regulators. They're like your cellular brakes. They help control when genes go on, when genes go off. And they do this by modifying protein called histones, which package your DNA. So when HDACs are active, they keep certain genes silent, silencia. Butyric acid attacks them. And so the butyric acid from PG inhibits or blocks HDACs from doing what they're supposed to do. So, like taking the brakes off of certain genes. In this epigenetic change, when HDAC is blocked, it leads to changes of how that DNA is packaged. That's what we call epigenetic modifications. So in this particular case, it specifically affects regions of the DNA called, this doesn't matter so much, but BZLF1 promoter region. That region normally, so it's taken the brakes off, right? This is the region that keeps Epstein-Barr in its night-night sleepy stage. So by blocking the HDACs, that butyric acid essentially unmasks this promoter region, making it more accessible. So while that BZLF promoter region is now exposed, it can get activated. The trigger of Epstein-Barr virus wakes up from its latent states and starts replicating again. That's how PG wakes up Epstein-Barr. So imagine it like this. Your DNA is an instruction manual. And so the genes are like individual instructions. These HDACs are like the cover on top of the instructions, it keeps them hidden. PG produced that butyric acid, acts like the tool to remove that cover. The cover is open. The EBV, the Epstein-Barr virus, gets the instructions open, and now the promoter starts, that's removed, and then it wakes up from its sleepy state. I thought that was just fascinating with PG. Am I the only one? And I'm not advancing my slides. There we go. So with the whole wrap up of this, we can see herpes viruses in the implication of oral diseases as well as brain diseases. We got to work together. We have to keep the research going. We have to keep reading the research when it comes out. We have to be educated of how to educate our patients. Right now, are we putting anybody on extra antivirals? No, but we can be perhaps testing for some of these viruses. We have to take cold sores more seriously because no other entity in healthcare is going to do it. And that's one of the best impacts that you can have for your patients, for their brain health. And that's probably about it. I want to thank you. I'm going to stop sharing. And if there's anybody still on this call, I really appreciate it.
And we're all still on this call. That was fantastic. Wonderful presentation. Very informative as always. Thank you so much. And we have a few questions. And anyone else that has questions, feel free to type them in the chat and we'll ask them for you. Or raise your hand and we'll unmute you and let you ask them live. But Christina Kraft wants to know, does shingles cause dry mouth or does dry mouth reactivate virus?
No, I would go the other way because there's salivary dysfunction with all of these viruses. So from everything that I read on every single one of these viruses, there was salivary dysfunction that could come from that. So it wasn't dry mouth going that direction. It was the virus affecting salivary glands.
Okay. Christina, anything else to add, or hopefully that answers your question. Alexandria Weiland says, is there any concern when like OB/GYN doctors put expecting mothers on daily prophylactic antivirals 30 days plus before birth if there's a history of cold sores in the mother? What are your thoughts on that?
They're doing it. They're not really doing it for cold sores for the mouth. They're probably doing it for genital herpes, because that transference and also cytomegalovirus can cause huge issues of birth defects. So maybe it is for cold sores. I wouldn't think a 30-day antiviral, especially in that case, is, I'm not an OB, but if it was me and they wanted me to do it, I absolutely would do it. There's no doubt, for sure.
Okay, thank you. Kathy Barth wants to know what dose is your husband on for his eye protection? Kathy has a dentist friend who has had numerous eye procedures and has had herpetic infections twice.
No, he gets cold sores. So he does famciclovir, and I think it's 1.5 grams once a day. There is nothing to do for his eye. He does not, I will say this, he only gets cold sores now. He has not had a flare up in the eye in forever. He gets very nervous if this goes to his nose on a location issue, and he's very protective about the whole thing. But to answer her question, not an antiviral for his eye protection. It's kind of shot already, but it is. He does famciclovir in, I think it's 1.5 grams, one big dose when the tingle comes.
Does he do anything differently? Like when you were talking about the plumes and things, like when he gets dental procedures to protect the other eye, or anything else you were saying he gets nervous when?
No, we're pretty solid. Those eyes are closed. He used to wear sunglasses all the time, partially because it was painful, but he's just really protective. He handles it. And I think if anybody in the practice has had that happen, they're going to tell you what they're going to do. You know what I mean? They're going to protect themselves.
Okay, that's great. I have another question, and it's that last few slides you went through talking about the gene expression. So I may have trouble articulating it, but what it made me think of is that the effect with butyric acid, because we're prescribing sodium butyrate or tributrin to support neuroplasticity. And when you went through that kind of fast, and so a lot of people are asking, is there a recording? We want to watch this again. Oh, we need to take, we're all going to listen to this again, or several of us.
I don't know if how that expression from PG, the way that expresses of waking up the Epstein-Barr to what you are putting, which I knew that you did that actually. I don't know. That does seem like a conflict to me. But in the PG world, and maybe it just has to do with that. I don't know. But I mean, butyric acid, I mean, I don't know. I don't know the answer. But now I have to ask Isaacson again. Thanks, Gina.
Oh, well, I'm going to go back and listen to this again and then study that as well, because it's interesting that we're thinking it's helping and maybe it's not. And I'm sure there's much more to the story. So I appreciate
think that there's far more to the story.
Very exciting, very interesting. It's very stimulating. I want to go dig into the science more now.
I'll
Not
send you those studies.
Okay. Thank
Well,
you.
I have two things. One, I guess everyone's getting for Valentine's Day some nose filters. We're going to be wearing nose filters during the day and our nose expanders at night. We're just going to all look so lovely all the time. Thank you for that, Anne, because that's all I could be thinking about is,
There's so much flu going around. It's crazy. Not that it's going to totally prevent the flu, but I don't know.
Lori, that's so funny because it makes me think, in other talks she talks about giving the blood pressure cuff as a wedding gift. Okay, and now we're going to give
Now we're going to be giving nose filters.
Valentine's Day. Okay, lovely.
That's why she speaks our language. It's like, what a great gift. And people are looking at it like you're crazy. But I have to say, as always, I learned so much. I have pages and notes here, even though you tell me I always learn something new. And I hope to all my saliva hackers out there that you're really taking some serious notes on the treatment. And we talked about this the other day. I know some people are logging off, so I do want to ask this before we get in this conversation. Where can people find you? If people want to learn more, want to know more, please tell us where we can go and learn about this to learn more from you.
Anybody can find me. It's always put my middle initial. So I have a website. It's annorice.com or an email, annorice@gmail.com. Just put an E on my name and an O in the middle, and I'm out there everywhere. I write articles for a magazine, and oftentimes it is kind of some outlier new things that are coming. It doesn't have to do with the brain. It's really more about oral systemic health and different antibiotics and resistance and all sorts of, that's what is kind of coming for this year. So there's a lot in that as well. So just if you have any questions or you want to tell me to get rid of the pink behind me, I've had more people than you say it. So
you're not getting rid of the pink. Anything, I just want everyone to know you have been awarded a distinction, right? You were awarded that by RDH. You do write for a column, and for all our fellow hygienists to be following you is so important. You and I have had collaborated on many occasions, and you are the person that I pick up the phone and say, hey, what do you think about this? I think we need more people in the world of research to be able to go to ask those questions, to know where to turn. But one of the things that we were having our coffee, I think we are having coffee and talking. I was having coffee. And we are just saying, why don't more of us treat at least HSV1? Why are we so, I think, fearful or afraid? And so that's where I was going with my saliva hackers, to just realize the importance. And you didn't talk too much about that statistic, but if we actually treat it in the office, can you tell us a little bit about the risk versus benefit, or the decreased risk to brain health?
I don't know, because lasers, and I'm going to say lasers are the most unstudied for efficacy. It is fascinating to me. We've had them for so long and they do so many great things, but there's not the huge compelling research. But here it is. If you can stop the replication, you're going to improve the healing. That's what an antiviral is for, right? That's what we're trying to do. We're stopping replication. If you're doing that with a laser, and to me, the way that I kind of look at it is the antivirals, when you get that first tingle within the first 48 hours, boom, great. Your antivirals aren't going to be all bueno if you've had that thing pop up and it's been sitting there for 3 days. The lasers, when it pops up, the antivirals didn't work or they didn't get them in time, so on and so forth. That's where we've got a sore, right? And we're going to stop it as fast as we can. So I think that there are two different timed entities, perhaps. You're going to take that as soon as that, I mean, you wake up and you feel something really weird, that's when it goes in your mouth, to try to drive down the replication. But lasers don't, drives me crazy. They don't have a lot. But how are we going to measure that? It goes into a latent, you're going to only do it by
If we give
healing.
somebody an antiviral, what is, I would say, the statistic on that? We know if they stop
Okay.
replication.
So I know it for Alzheimer's disease is this: when we don't treat with antivirals, it's a 50% increased risk. Now, remember, that doesn't mean, remember what 50% risk actually means. Okay. That doesn't mean that half of everybody that gets cold sores are now going to get, that are going to get Alzheimer's disease. But what we also know is when we put people on antivirals, not only is that gone, we are actually reducing the risk. It's actually a risk reduction. That's huge. And I will say that it, just my opinion, I think in the future, and this conversation came up when I was there, is that we are going to be really looking at antivirals in a cognition world in the future in our older populations. It will be something. You know, way back in the day, does anybody remember that? Antivirals were given as a preventive prophylactic drug. That was for years. I just heard a pharmacist say it the other day. I the people that I talk to, we're not quite there yet. I mean, and I'm not your doctor. I don't necessarily would recommend that. By the way, you're going to get cold sores after you turn 35. Your cold sore drops dramatically. So is that a hormone thing? Why is that? I don't have the answer to that. But as we get older, we have, for the people that get them, they have less reactivation. So I don't know.
Yeah, and my point to that is just not being so hesitant or afraid. I think for
Printing an antiviral, why would you be I don't understand why dentistry would ever be hesitant about that.
I think a lot of people are. They're just very afraid to write for an antiviral. I don't know. I'd like to see maybe raise the hands, or people are say, yeah, hesitant or don't do it on a regular basis. And that's really been one of my takeaways is, to spread your message and not to be so hesitant, right? That there's such a benefit there. There's another question from Cassandra. She says, for patients that are testing positive for multiple times this winter with the cytomegalovirus, though they're asymptomatic, what would you recommend?
For them to talk to their physician, because we're not going to do an antiviral for cytomegalovirus. So I mean, that isn't the protocol that most doctors, what is their risk? Where are they? Are they immunocompromised? There's a whole bunch that goes into more precision medicine in that. Do they happen to be getting sick? Are they immune compromised just with, are they stressed? How is their health and all that? But that's where I lob that back to a doctor. Because with that's not a common antiviral. Neither is Epstein-Barr.
Yeah, and so we talk about that a lot, just opening that conversation, like you're saying, talking about their immunity, right? We know it can exacerbate the periodontal. And so I think those that are comfortable with talking about supplementation and immunity and sleep and all those things, like you're saying, stress, having some of those conversations. But right, somebody who keeps expressing, we need to probably get them, and I say we all say that, get them in the hands of their physician, so somebody's keeping an eye on that. So love that.
What did
There
Catherine say that Doug Thompson teaches?
just our good friend, Dr. Doug Thompson, who teaches also, is just in alignment with us. I think all of us are in alignment that he says testing for viruses and prescribing antivirals. So I think what you're hearing across the board is, yeah, all of us are in agreement. I don't think any one of us in this world would say not to. I think we just tend to find that people haven't done it as a regular habit. And so love hearing that. Well, any other questions? Anything anybody likes to raise their hand, drop in the chat. Getting lots of thank yous. Can't thank you enough, Anne. It's been absolutely fabulous. And because of where we're going with brain health, and like you said, that we're only going to get more information, we're definitely going to be having Anne back on. She is part of our community, and we appreciate you so much, value all that you bring to this community, and look forward to hearing so much more. Dr. Gina, what do you have to add with all this great information?
I just want to thank you as well. Yeah, opening this conversation up tonight will lead to a lot of stimulating conversation moving forward, and it's very important points that you've made. We all look forward to looking at the presentation again a little more slowly, adding to our notes.
Great.
Thank you so much. Great
They'll
information.
find all the flaws in it. I'm just kidding. And if anybody wants any of the papers that go with some of those slides, just send me an email and I can, some of them were at the bottom of the slide, some of them weren't, but
And we have
happy to
one
do that.
question. Can we take this, Laura? It's time, but anybody that wants to stay on, Christina raised her hand. Christina, what would you like to ask? Just go for it. She's unmuted. Then did we lose her? I think, oh, go ahead, Christina. We can't hear you, but I think you're unmuted.
Yeah, she's unmuted. We can't hear you. I see your lips moving.
Did you put your question in the chat?
Yeah, can you hear me now?
Yeah. Yeah.
Great. Okay. My question is, I recently had training in dental ozone therapies, and one of the things we were talking about was its use in treating cold
Yes.
canker sores, and I was wondering what your opinion was on that.
Okay. I know that Kelly, in not theory, that it would make perfect sense. I know that Dr. Kelly Mish up north has done it for a long time, and she probably has lots of before and afters. I reached out to her once and I didn't hear back, but she's used ozone on cold sores for a long time and she continues to do it. So then I would see efficacy with that, or she wouldn't do it. So yeah, absolutely.
I was wondering if it has, I know what I learned with lasers is, maybe this isn't right, the light energy actually travels up the ganglion and can reduce the occurrence if used often in the same expression. I didn't know if ozone is just
I don't know. I don't know.
Okay.
It's funny because when she was one of the first ones that I heard that would start doing it, but reoccurrence, how do you ever measure that? We're never going to know if it would just be somebody's history. You know what I mean? That would be tough. If it works that one time, then keep doing it.
Okay, thank you.
That would be all that would matter.
Thank you so much. That was amazing.
Thanks.
Well, great community. Thank you so much. Please go visit her oral systemic seminars there at annorice.com. Appreciate it. Love you for giving all this great information. And our next meeting, remember, we go every other month, will be in April on April 2nd, that first Wednesday of the month. And we're going to be talking all things AI. So get ready for that. We are, yes, heading into, as we start 2025, we're going to be staying on top of all things that are current. So I look forward to seeing everyone there. And thank you. It's great seeing everyone's faces tonight. Have a great night.